It's my great pleasure and honor to introduce Susan McCouch, the Barbara McClintock Professor of Plant Breeding and Genetics Emerita in the School of Integrative Plant Sciences at Cornell. It's so appropriate for us to be hearing from Susan, because the Barbara McClintock Chair, because Barbara McClintock, of course, is one of Cornell's most famous scientists. She, in the 1930s, made great advances and, in fact, won a Nobel Prize for her work in corn genetics. Today's subject is rice. Rice is a food staple and has been for well over half of the world's population. And it's culturally significant, and it has tremendous economic impact in national economies, world economies, for millennia. Susan will introduce you to the diversity of rice as seen through her eyes as a rice geneticist and plant breeder. Susan is a homegrown academic star. Her research on rice began as a graduate student in biochemistry and plant biology in the mid-1980s, and she got her PhD with Steve Tanksley and Ronnie Kaufman in 1990. She did a postdoc for 5 years in the Philippines at the International Rice Institute. And after the postdoc, she returned to Cornell, and for the next 30 years, she's been here. She is known for developing the first molecular genetic map of rice and for her role in turning rice into a modern system for genetics and breeding research globally. Susan's work is of international renown and has earned her academic recognition, such as election to the National Academy of Sciences, along with many, many national and international awards, prizes, and honorary degrees. Susan is truly one of Cornell's great distinguished scientists, and she's here to tell you about her life's work. Susan?
Thank you for that lovely introduction, Ron, and it's nice to see so many familiar faces here on this snowy winter's day. And let me note that when you are the recipient of many international awards, sometimes it's because the work you do has absolutely no significance locally. So one of the great, dramas about hiring me when I joined the Department of Plant Breeding at Cornell was that plant breeders at Cornell were paid by the public university, by New York State tax money. And how could you possibly rationalize the hiring of a rice breeder in the state of New York? So we are sort of going to come around full circle a little bit about why some of the work we did, for me anyway, has turned out to be quite significant here, and largely because the molecular biology revolution transformed our understanding of biology in general. And it allowed us to intersect species in a way that we never could have before. So I've titled this, What's in a Grain of Rice? And of course, the answer will be different for each person, but I will take you on a little tour of some of the things that I've discovered about rice that have made it a world of investigation and discovery for me. It opened up a part of the world that is mostly Asia and parts of Africa that I had never been to, and transformed my life in many ways. I'll start with a very fundamental question that many people ask, and some people probably already know. What is the difference between brown rice and white rice? Are they different varieties? Do we breed for brown rice separately from white rice? Well, the answer is no, they're genetically the same thing. White rice is polished brown rice. So brown rice has a layer called the bran, the rice bran. It's 2, sometimes 3 cells thick. It's a very tiny, thin layer wrapped around the endosperm and the germ, or the embryo that will give rise to the next generation. That wrapper is the female's ovary. It dries up and holds the germ tightly adhering to the endosperm, the starchy endosperm, which is mostly what you eat when you eat rice. When you polish off the bran layer, you get white rice. But what happens is during the polishing, which is kind of a sandpaper-like motion in a larger area, that little rice germ is often lost. And so if you look at your white rice, it has a little missing edge on one corner. That's where the germ has fallen off. So if you're a farmer, you want to plant the seed, which I show just above the polished white rice there, which has the hull on it. And it contains the wrapper, which will ensure that there's an embryo in there that can sprout. And it will use that endosperm, that starch, to feed the developing embryo. So that starch, which is what feeds us, is also what feeds the growing plant. If you also know a little bit about brown rice and white rice, you know that there are two major differences from the culinary perspective. One is that brown rice is more nutritious than white rice because those two cell layers that we polish off when we get white rice are full of vitamins and minerals, a little bit of fat, a little bit of protein. So when you polish them off, you remove that part of the nutritional complex, which is the rice grain that we eat. And so then you might ask, why do people polish off the most nutritious part of the grain? And there are two answers to that I think many people may not appreciate. One is that we polish that bran off because in the tropics, brown rice does not keep unless it's refrigerated. It goes rancid very quickly because of that oil and mineral and vitamin content in the bran. So if you want to be able to keep rice from one season to the next, or transport it, or keep it in your granary, people learned long ago that if you polish off the bran, the white rice can keep for years. And so the real reason that we think about that as a negative is because we think, well, people probably just throw that bran away. They just discard it. But no, of course they don't discard it. The bran is polished and it's a valuable marketable product, which is used in cereals, in cooking, and baking. Sometimes it's fed to animals in any case, and it would be the thing from which we derive rice oil and some of the other byproducts. But most of the time, the bran is just kept and sold separately. Sometimes people sprinkle the bran on top of their white rice. So bran is not wasted. And the second reason people like to polish rice, especially in the poorer parts of the world, is because if you have to walk a long way to bring water and fuel, you know that white rice cooks in half the time. So it takes less fuel and less time cooking and less water to cook your rice. And that saves many, many hours for the women who cook the rice. So there are lots of reasons why people learn to polish their rice, save the bran, and eat the white rice as white rice, which is what most people in the world do today who depend on rice. Now, rice production per country. This graph shows that China and India, the two largest, most populous countries in the world, produce the most rice. In fact, they produce about 4 times as much rice as any of the other countries. The top 10 countries in terms of rice production are all in Asia, followed by Brazil in South America, then Japan, then the US, and then Nigeria. And I go down to those 14 just to include all the continents. So you see that the US is not a major rice producer, but it's in the top 14. It's not in the top 10. And the rice that we produce, the rice that we sell commercially, comes from Asia originally. We breed it to be adapted to the subtropical environments in southern US and in California, but we do not, by and large, commercially sell American wild rice, and I'll get to that in a moment. So that's the rice producers. What about the rice exporters? Well, the major exporters are only these 3 countries, the largest being India, that exports mostly its supreme quality basmati aromatic rice. Second is Thailand, which produces something that many people know of as Thai jasmine. Cooks up very differently than the basmati, but has a very distinctive, lovely aroma when it's cooking. And then interestingly, Vietnam. So for those of you of my generation, we remember what was going on in Vietnam when we were young adults. It was a horrific scene, and much of their rice production was destroyed by Agent Orange and other things. But they bounced back, and they're now in the top 6 producing countries of rice, and they are the 3rd largest exporter. What they export, interestingly, are long grain white rice that is broken into tiny pieces. And it's sold as pearl rice so that it cooks up in a very different way. And who would have thought that you could get a market niche by breaking long rice kernels? But they did it. Global rice production has quadrupled since the 1960s, which was when the Green Revolution began. And the International Rice Research Institute was the home of the semi-dwarf breeding project in rice. So I'll be talking a little bit about the International Rice Research Institute, but not about the Green Revolution. Excuse me, because I came in late for that. But the point here is that here you see this graph, and there are 2 major reasons why we've been able to quadruple rice production and yields globally over the last 60 years. And that's a combination of breeding and management. Management in terms of water, the form of irrigation, in the form of fertilizers, both organic and non-organic, and pesticides, which, by the way, have dropped over the last 60 years. We use less pesticides today on rice than we did in the 1970s and 80s, thanks to the breeding effort that is kind of caught up with putting disease and insect resistance genes back into the improved rice varieties that are so high yielding. The other thing we've really done is improve the grain quality of rice globally, because the Green Revolution rice, although it produced a lot of grain, the quality for humans was not very high. And I just make this point because if you look at the yields of corn or even wheat, the yields have gone up in corn even more than in rice over the last 60 years. But most corn is not eaten by humans. It's fed to animals. And they don't care so much about the quality of the kernel. So what is Asian rice? Asian rice is a perennial. It remains a perennial today. It's an annual because if you plant it in a climate where there is winter, the winter will kill it. But if you plant it in the tropics, it will grow and grow and grow. If you cut it back, it will resprout. So it's a sort of an interesting feature of rice is that it remains a perennial. It's a semi-aquatic plant. Its ancestors, all wild ancestors, all grew in aquatic areas. And it's grown on 10% of Earth's surface, one of the most extensively planted crops in the world. In the tropics, it can be grown 2 or 3 times. You can get 2 or 3 plantings per year. In the temperate zone, we get only 1, but in the subtropical zone in the southern US, for instance, we breed for a rice that can be harvested in September or October, and it will retune back. It will give you a second crop without harvesting the plant. What you cut down when you harvest the first time will grow back, and they get what we call a retune crop. Harvesting that retune crop is the difference in the southern US pretty much between break even and making money for a farmer. So breeding for that perennial quality is incredibly important in the southern US today. What I'm showing here in this picture, for those of you who can't quite imagine it, is a row of rice workers in India. In this case, the women are all doing the transplanting. There's a male at the end telling them what to do. And you know, this is the way it is in much of the world, but they have transplanted that rice that you see in the foreground, and there's a patch in the background that's perfectly rowed out. And on the left-hand side of this slide, you see their little bunches of rice. Those are the packets of germinated seedlings that they're going to then take and put one by one in place as they plant into this muck soil. The other thing I think you should notice is they're up to their knees in muck. They're not up to their knees in water because the rice paddy is prepared for weeks before it's time to transplant. And so what you have is a murky kind of clay soil that we call it paddy. It's been worked over with a water buffalo or a small tractor to create muck. And when you stand in it, you sink down up to your knees, but then there's a, oh, sorry about that. But then there's a hard pan below. So these women are transplanting into very soft soil, and they just throw the rice in, and the roots will accommodate down, and the stems will start to grow upward. Most rice, I've given the term, I looked it up, is 82% these days, is produced on small farms, mostly in India and China, as you've seen, but throughout Asia. A small farm is categorized as something that's less than a hectare in size. So believe it or not, today, unlike maize or wheat or soybean, this rice is still produced this way on a majority of farms that are less than a hectare in size. And over 90% of rice that is grown is consumed in the country where it's produced. Again, it never hits an international market. Nobody knows about the varieties that are grown internally because they are never traded very widely. So rice remains very local, and that means that there are special culinary flavors and tastes that are evolving with the rice that people grow in their own villages, in their own cities, in their own provinces. And most of us never see that great variety of rice that's actually planted. I mentioned that rice is also native to Africa. Oryza glaberrima is the species and genus, the genus and species name for the rice that was domesticated independently of Asian rice in West Africa, as I think you can see on this little map of Africa. It has typically red pericarp, so the bran, rather than being light brown, is quite red. And this beautiful hue that you see here gives it a series of interesting flavors if you cook it as red rice. It cooks up with a kind of nutty flavor because those vitamins and oils and minerals in the bran convey a certain flavor and quality to that rice once it's cooked. It also has a slightly different hull color. You see from this photo, it's got dark hulls, and it doesn't have a branched panicle, so it's not as high yielding as Asian rice. We also have rice native to North America. Many of you know it as American wild rice. It has a different genus and species. It's not crossable with Asian rice, but it does have the same number of chromosomes. It has an ancient ancestor in common. We call it Zizania palustris in terms of its Latin name. It's a sacred food that was collected by indigenous people, including the Guayacono people of the Cayuga Lake region of New York. So it's native to this area. And you can see on this map, it's very commonly found even today along all of the Great Lakes. And early in our history, it grew all the way down the coast, down into Florida, Georgia, and Louisiana, wherever there was swamp country. So because it is an aquatic plant, any of you who are familiar with the swamps down in that area, that was the original home of the wild rice. And under the slave economy in the early days of our republic, we had the importation of rice from Asia and from Africa, which was then planted in those paddy areas, those wet areas. And many of those swamps were drained in order to support rice production in the southern US. It's important to talk a little bit about ecotypes. Ecotypes are genetic adaptations to different environmental conditions. And the ecotypes of rice are defined largely by the water table. We have dryland rice and wet rice. Okay, so some rice is grown, planted on dry soil above the water table. For instance, the upland rice is grown on hillsides. The rain-fed lowland rice is planted just at the water table, and we actually breed for thick, strong, deep roots for the rain-fed lowlands so that it will penetrate the water table and access water below the soil. Then there's the irrigated paddy, which is the one I just showed you in the picture of the women transplanting. And then there's something called deep water rice, which is a very unique ecological niche. We find deep water rice growing in areas where there is seasonal flooding, mostly in Thailand, India, and Bangladesh. And these are regions where the flooding is every season and it comes at a predictable time. So they plant the rice before the floods come. And the adaptation that these rice plants have is that as the water rises, they elongate their stems to keep the growing point always above the water. And then those rice plants are harvested by boat, much like indigenous Americans harvest the wild rice in this continent. This shows the land mass that's planted on a global basis to these different types of rice. The irrigated paddy, which is the one that most of us think about when we think about rice growing, accounts for 50% of the land, while rain-fed lowland accounts for about 36% of the land. But in terms of production, that irrigated paddy produces 75% of the rice that's harvested on the planet, while the rain-fed lowland accounts for only 19%, and the upland and the deep water account for very small fractions of what is grown. But the point is that there are regions of the world, there are ecologies in which no other plant will grow, except these particular rices that are adapted to these hydrological conditions. Most crops cannot grow in standing water. Rice is very unique in this way. So I want to talk briefly about each ecosystem. The irrigated paddy system is a very productive, as I've just explained, highly productive use of very wet land where other crops won't grow. It's the most ecological form of weed control we know of. If you spread water on a rice paddy, what you do is suppress all the weeds that would grow in dry land. But since rice can survive and most of the weeds can't, that water is a lifesaver for people who never had herbicides and didn't want to go out and pull weeds all the time. It also provides optimal nutrient availability because in a saturated soil, an anaerobic soil, the pH goes to neutral. And at pH 7, all the nutrients that the rice plant needs are very available. They are easily accessible to the rice plant. The rice plant also has a unique anatomical set of tissues called parenchyma. And it's basically like a straw that goes from the leaves down the stems all the way to the root. And it allows for gas transport. So when a rice plant is growing in saturated soil where there's no oxygen in the soil, it still needs oxygen in its roots. It gets it by sucking on this straw, and it draws oxygen down from the atmosphere and feeds the roots. And the problem is not that that isn't wonderful. It's terrific. It's the reason why the most wet, let's call them water-loving plants can live in the saturated soils. But down in the roots, there are both beneficial microbes and also the microbes that produce methane. And that little straw also allows the methane to move through the soil particles and be back up into the atmosphere. So the straw-like function of the parenchyma is a gift when it comes to rice growing in saturated soils, but for global warming, it's allowing the methane to come up just like candles on a birthday cake. For every rice plant in that paddy, there is methane flowing. So we have problems with the paddy system. Wonderful form of weed control, but in order to stop the methane from being emitted from these soils, what do? You pull the water off. You drain the water during the mid-season, and then the methanogens can't compete with the oxygen-loving bacteria. You get a preponderance of the beneficial microbes in the soil. You push back the methanogen populations, and then you bring the water on as the rice is trying to fill later in the season. And this is now being practiced globally. In fact, it's being mandated by most governments. It's called alternate wetting and drying, and it allows the rice paddy to become a less harmful system. Rain-fed lowland has numerous problems related to water. Sometimes it's drought, as you see in the upper left corner. Sometimes it's flooding, as you see in the lower right corner. And sometimes because there isn't standing water on these soils in the rain-fed lowland system, what you get is a pH that goes to very acidic. So it goes down to about 5, pH of 5. That creates massive nutrient disorders for the rice plant, because at pH 5, even though it can access water deep down under the soil, for instance, this iron toxicity, you see the red soils there in Colombia. You also see red soils like that throughout much of West Africa. You have the acidic soils, as we're showing in the picture from Liberia. Rice plants simply crumble under that kind of stress. But then when the water comes, they can grow again. So the rice plants that grow well in rain-fed lowland have to have diverse forms of abiotic stress so they can withstand all kinds of drying and flooding and acid soils and other problems. And these plants are very resilient, but they tend to be lower yielding. Upland rice is still grown in certain parts of the world. It's a small percentage now of what is grown, but you know these things as slash and burn agriculture. This photograph shows a hillside that's been where the forest has been cleared so that for 2 or 3 years you can plant crops. They're normally not planted as a monoculture. It's planted intermixed, rice with other things in these highland tropics, if you will. And what I want to show with this picture is, you see this picture, these little green things sticking up are the little rice plants. So when rice is planted and it grows on these soils, the first few years after clearing the forest, they're very fertile, and the rice plants grow pretty well. And the beauty is that these plants have all kinds of unique genetic attributes that breeders can use to help improve the plants that we've already produced under, previously, to find traits that give special resistance to some of the soil problems that people encounter. So these are very unique seeds, and they've been selected over millennia by people in mountainous parts of the tropics. And finally, there's deep water. If you've never seen deep water growing, what you see in the lower left is a breeder standing up to his neck in water and clutching a plant to see if it's firmly rooted. Because these plants, unless they're firmly rooted, they'll just move with the water. And then in the middle, you see a couple of women out there. They're doing their evaluations and they're bringing samples back. And on the right-hand side, you see what would happen if you drained that flooded area, how tall the rice plants have grown, 12, 15 feet high. Now, of course, they wouldn't stand up if you drained the water, and you don't normally drain the water, you harvest in the boat. But that's how elongated these rices can become. And they have a very unique feature. And for those of you who are interested in genetics, we worked with another group and we cloned the genes that are involved both to make them semi-dwarfs, which is the productive green revolution type. And those are very short plants. And these that elongate up to 15 feet high. And it turns out it's the same gene. It has a different promoter. It responds to different environmental signals. But it's the gene that tells the plant whether to elongate or not. And when we cloned it and showed that it was the same gene, I can tell you it stunned the world. So the very gene that gave us the green revolution has also given the Thai, and the Indian and Bangladeshi breeders this elongating rice. Now let me say there are seeds of 180,000 different diverse accessions that are preserved in the Erie Gene Bank. It's called the Genetic Resources Center in the Philippines. The building is shown there just to make you pause for a moment and ask, in a country that suffers from earthquakes, cyclones, tornadoes, typhoons, why would you put the gene bank there? Well, they built an earthquake-proof vault underneath in the basement of that building where I used to work. And in that earthquake-proof vault, there are backup electricity generators so that during the cyclones and times when there's no electricity, which happens almost every day in the Philippines, even the hospitals will not have electricity, but the Erie Gene Bank will have electricity. Because if those seeds thaw, the world's largest single species collection of diverse seeds would be lost. Now, what is in there? Well, it's both wild and cultivated rice collected from all over the world. It serves the purposes of conservation of that diversity for seeds that may no longer be grown by farmers, for safekeeping. Many stories, some of you may know them. For instance, after the Vietnam War ended, Cambodia had lost almost 90% of its native varieties, and they were able to be retrieved from the Erie Gene Bank and repatriated, or rematriated, as we like to say, back to the country of origin, so that on its recovery after the war, they could plant their own varieties and proceed. It's an astounding thing to realize, what a gene bank can do. It's also the foundation for breeding and research, which was the reason I got to use the Erie Gene Bank after all. So you might ask, how many varieties are actually grown today? We have a lot of them in the Erie Gene Bank, but how many are grown today? And the answer is, we don't know. We have no idea. There are thousands of locally adapted varieties grown in the hills and the valleys of the Himalayas and across the floodplains of Asia. And many of them are shared only through convenios, what do we say, alliances like marriage, through kinship relationships. Some are shared through trade. Some are kept as sacred ancestral varieties within a village. They will never be deposited in a gene bank. But every country has a national gene bank for the types of crops that are important to that country. And then there's this system of international gene banks, and Erie represents the home of the gene bank for rice. This is what the Genetic Resources Center looked like in 1970, when farmers and collectors started sending packets of seed just in paper bags with a ticket on them. And all the ticket said was the name of the variety, the place where it came from, and occasionally what that variety was known for. But believe me, these tickets were handwritten in the 70s. They were written in Thai, they were written in Hindi, they were written in Chinese, and they came into the International Rice Research Center in the Philippines, and they had to translate all that information from those handwritten tickets and start to keep a database. So it was a challenge. By the 1990s, when I arrived, the effort had transformed from trying to translate passport information on seeds to trying to keep seeds for the long term. And all the seed packets had been regrown, and the seeds had been harvested, and they were put in vacuum-packed aluminum little packages and stored in what looks like the kind of library that we think of for a modern library, with stacks that move. And you'll notice these people are wearing their overcoats just like we would on a day like this, but you're in the middle of the tropics in the Philippines. It's because it's -20 centigrade in the gene bank. And that's why those generators are so important. By the 2010s, what we start to see is that people are able to access information in the gene bank online. And I get ahead of myself here. I'm going to go back for a moment and just say, the reason I wanted to show this slide is because I had a small part in helping transition from the concept of genetic resources as simply a physical thing to thinking of the concept more like a library where you could search for information. My undergraduate degree is in literature and history, specifically Hispanic literature and history. I spent many years in Spain. I lived there under Franco. I had a kind of an interesting undergraduate career. But when it finished, I had a strong interest in understanding what was going on in the Hispanic world in the Americas. And so in 1976, I undertook a hitchhiking trip with my partner at the time, and we went from Boston to Buenos Aires by land. It took about a year. We were not in a rush. We were interested in seeing all kinds of things along the way. And during that time, some of the things that really caught my eye totally changed what would be the course of my life for the future. I noticed in the tropics how people multi-cropped and what the crops were. and how people interacted with both the natural world and the plants that they cultivated. I saw in the deserts in the high Andes how water would transform what was basically a pretty infertile looking place into something that could produce abundant food. And when I got to the south, it started looking a lot like the north, and I felt quite at home. And so, in fact, I lived down in southern cone of South America for several years before returning to the US. And I'll just jump ahead. I did a master's at UMass. I met the man who would eventually bring me to Cornell. And as it turns out, at Cornell, I was able to join the Department of Plant Breeding and Genetics for my PhD with Steve Tanksley, shown here in 1986. It turns out that when you join after you've been out of school for a long time and you had a very different career to begin with, you're a lot older than most of the students. And it turns out that my advisor, Steve, and I had been born the same year. So my great professor advisor and I were contemporaries, and we shared many, let's say, perspectives on life. And I felt very fortunate to be in his lab. There's myself as a young scientist in '86, and we were joined by two young Chinese scientists who were some of the first people to leave China after the Cultural Revolution. They arrived at Cornell in 1986. We were funded by the Rockefeller Foundation to develop the first molecular map of rice. And my Chinese colleagues knew a lot about rice. Steve and I knew a little bit more about molecular biology. Together, we were an awesome team. And in 2 years' time, we created the first RFLP map of rice. Those of you who are geneticists will remember the days, southern analysis and radioactive probes and very tedious work. The way this worked 24/7, because we were in a big rush, we were in a head-on competition with the Japanese who were trying to also develop the first molecular map of rice. And Steve said, let's see if we can get there first. And I already had a young baby, so of the three of us who worked in the lab every day, they gave me the 8 a.m. to 5 p.m. shift. When I would go home at 5 p.m. to pick up my daughter, then Zhang Yuan Wang would come in. He would serve from the 5 p.m. to midnight shift. And when he finished, Zhihong Yu would come in at midnight and serve until I got there at 8 a.m. And we worked round the clock with my Chinese colleagues, sleeping in the same bed. One would get up and the other would go back to bed in that bed because they were here under very sparse conditions, let's say, in terms of what they had to live on. Why have I circled 3 red circles there? I just wanted to remind myself to tell you that during the time we were developing the map, we discovered the first active transposons in rice. And this was Barbara McClintock. I'm the Barbara McClintock professor, after all. It was Barbara McClintock who won the Nobel Prize in 1983. This was in 1986. She was hot on the news, and we discovered the first active transposons in rice. And those 3 elements were all moving, actually actively moving in the population we were mapping. The blue one is the first gene that was ever cloned in rice. We cloned it, but we didn't know what it did. So it wasn't big news. It was just a marker on our map. All right. So if you've worked in this kind of science, the question is, everybody was mapping the genome. The human genome wasn't yet mapped. There were animal genomes being mapped. And we were among the first that created these maps for plants. In fact, mine was the 3rd RFLP map ever developed. It was big news. And it was a hot opportunity because we published before our colleagues in Japan. But the reality was that a rice map wasn't going to do us much good in New York State. So I got my PhD in 1990, and I knew what I wanted to do with that map. I wanted to go to Erie and help put that map to work in the breeding program. So here's a picture of the International Rice Research Institute with all those rice paddies in the foreground and those beautiful volcanic mountains in the background, and the institute off where those palm trees are. We put together the first molecular lab at Erie, had to develop all the permissions for radioactive probes and all the stuff that goes along with it. It was quite a tedious job, but we also trained a large group of young scientists who wanted to bring molecular technology into the breeding program. And knowing very little about rice, I just took my cues from the breeders who were already there. They, after all, had been working on rice for eons. So they told me, well, there are 3 groups of rice. There's the Javanica, which derives from Indonesia, the Japonica, which comes from Japan, and the Indica, which comes from India. Funny, nobody thought that rice came from China. Why was that? Well, think about it. When Erie was founded, the Cultural Revolution meant none of the Chinese scholars were able to travel to the West or even into the Philippines. So China was almost not represented in the context, the cultural context of the International Rice Research Institute. Hard to imagine how different times were. Their classifications were based on things like morphology, grain shape, cooking quality, geography, and then crossing barriers. But I was there with a brand new technology, and I thought, great, I'll go into the Erie Gene Bank, we'll analyze thousands of the varieties that are there, extract DNA, and do some molecular analysis, and then I'll know which variety belongs to which of the 3 groups, and we could add it as information in the gene bank files. Wouldn't that be exciting? Well, sure. And this is what the principal components analysis told us. We saw 2 ancient groups based on chloroplast DNA, which some of you will understand. These are matrilineal groups, so you can trace back your origin to Eve. If you look at mitochondria in humans, well, we use chloroplasts and we can trace back to early maternal ancestors. It's maternally inherited. The 2 ancient groups were Indica and Japonica. All the rices belong to one or the other. We also saw 5 derived populations: the Aush, the Indica, the temperate Japonica corresponding to Japan, the tropical Japonica corresponding to Javanica, to Indonesia, and something called Group 5, A for aromatic. But it was just called Group 5 because we didn't have any idea molecularly what that little group of purple dots was sitting in the middle of our PCA. But they were clearly, they had a Japonica maternal ancestor. The fact is that when we looked at the 3 groups that we've been talking about from the Erie breeders, Indica was Indica, temperate Japonica, as I said, was Japonica, tropical Japonica they called Javanica, but they hadn't really accounted for the other groups. What they did tell me at the time I presented this information, they said, oh, no, you're wrong. These 3 groups are all Indicas. Aush is not anything, it's just an Indica. And that thing you're calling Group 5, it's just an Indica. I said, oh, really? Well, genetically, they're quite different. They cluster very differently. No, they said, see, those are all the slender grains. So those slender grains are those are just Indicas. And the ones with the rounder grains are Japonicas. That's how we know. Oh, I said, that's interesting. And I had a little bit of a problem. I started mapping our genetic groups back onto geography. And sure, we do see an east-west divide with the temperate Japonica and tropical Japonica on the east. That Japonica clade is called Geng in Chinese. And the Indica and the Aush on the west, if you will, side of Asia, called Indica or Xian. And then there was this funny group called Group 5. And I couldn't understand, it wasn't east-west divide. We were claiming that these were Japonicas, but they were way on the east. So the question was, how do we explain the Japonica ancestry of these Group 5 rices, which turns out have a special name? They're known as the Basmati rices. Somehow, a ancient Chinese variety had been transported all the way over to the far northern Kashmir province of India and up into the Pakistan and Afghanistan there. And I said to them, something must have happened. How how else could we get this result? They said, you're wrong. No, Basmati's Indian, therefore it's indica. Okay, fine. This is a national point of pride. Who am I? A young Caucasian scientist, never having lived in Asia before. I bow to you, I must be wrong. But looking back, it turns out that the genetics documents ancient migration and hybridization of rice along the Silk Road when back from 2002 BC to the 1450s. And this Silk Road was used, and many goods and technologies and knowledge and even religious and academic or intellectual things were traded and shared along this road. And if you look at the Silk Road and you map back out those genetic groups, what you see is that the Silk Road dropped down into different places along the areas where those early rices were being developed. And what we can now show genetically is that Basmati is actually resulting from crosses between Chinese Japonica and the Aush subpopulation, which gave rise to what is now considered native to the Kashmiri provinces in India. And I just remind you, Marco Polo traveled the Silk Road from 1271 to 1295. And along that same road, he carried Chinese rice all the way to Italy. And it is the foundation of what is today risotto rice. So we weren't so far off. Now you'll know that rice is primarily, how am I on time? Oh dear, I have some stories to tell. We can share those rices if you want. I brought in some little samples because I'm going to talk a little bit about some things that I thought it might be helpful if you just took a look. They're all packaged in closed plastic things. You can take them up, shake them, they won't fall apart or open. And just to give you an idea of what we're talking about here. So rice is eaten primarily as a whole grain. Unlike maize or wheat, wheat is usually made into flour, made into noodles or cookies or bread. But rice is eaten and appreciated as a whole grain. So the size, shape, color, texture, and aroma of every grain is what gives it its value in the marketplace. And fragrance in rice is rare, but it's very highly prized. So my group started to ask questions. I'm back to my Basmati group now in India after they finally agreed that maybe rice was native to the Kashmir province, but that it had arrived there through this nefarious means of hybridization and interesting gene flow. And so they wanted to know what was the origin of fragrance. And the Indians were sure that they were the origin of fragrance in rice. So let me just say, fragrant varieties have a very strong aroma. It smells something like popcorn, buttered popcorn. The major fragrance compound we refer to as 2-AP, 2-acetyl-1-pyrroline. And the gene that mediates the 2-AP production codes for an enzyme, which is betaine aldehyde dehydrogenase. But for those of you who don't want to remember such a long name, just think of it as bad H2, okay? The bad H2 gene mediates the accumulation of 2-AP, which is the molecule that we sense as fragrance. It has a very low odor threshold. So a human can detect that fragrance molecule at a density of 0.1 parts per billion. Aroma is produced in the grains, the leaves, and the stems, but not the roots. Most of us buy our aromatic rice and we only sense it when we cook it. But if you walk through a field of Basmati rice, just walk through the field, the aroma is in the air. So aroma is a really special magical thing for those who like aromatic rice. And I asked the question, why are rice varieties fragrant? And I wanted to understand it because if we knew that, we could use that as a tool to breed fragrant rice varieties more quickly and more easily. And wouldn't everybody in the world like to have aromatic rice? Right. So this is what the BADH gene looks like, BADH2 gene looks like. If you're not a geneticist, don't worry about it. It's not important, but it's a big gene. It's 5.8 kb and has 17 exons. A functional BADH2 gene inhibits 2AP production and gives you non-fragrant rice. The vast majority of rices are non-fragrant, and some parts of the world don't like fragrant rice at all. They find it offensive. For instance, southern India. If the BADH2 gene is mutated in a certain way that knocks out the function of the enzyme, then you get fragrant rice. So fragrance is a derived form due to a mutation in the BADH2 gene. The mutation that we were able to identify based on sequencing of these genes is an 8 base pair deletion in the 7th exon, if you will, of the BADH2 gene. And then when it's knocked out because it has that 8 base pair deletion, we give it a small letter. Instead of the capital letter, we call it BADH2.1, meaning it's an allele for a knockout, okay? So we identify the allele, the variant of this gene, as a small BADH2, and we give it 0.1, because there might be other mutations in the gene we don't know about yet. And that's what allows fragrance to accumulate. So then we asked the question, we had all these varieties from the Erie Gene Bank. Do all the fragrant varieties carry the 8 base pair deletion in the BADH2? That would have been a nice story. I could see, fantastic. It's all black and white. You have 8 base pair deletion, you're fragrant. So what we did was we sequenced, turned out 266 diverse varieties from the Erie Gene Bank, and we compared sequences. Now, this is a little bit much. Let's just say these are the countries in Asia from where we found varieties in the gene bank that were fragrant. And this is going to be the diagram that shows us where the mutations are. And I'll just start by saying, on the left-hand side, that's the BADH2 allele. So you can see if you look down that there are actually now 10 alleles known. This was the discovery we made by sequencing these genes from the gene bank. Mutations are diagrammed as to which exon, where they occur in the gene. The subpopulation, meaning which genetic group the variety belongs to in which these mutations have been found, and then how much of the aroma 2AP is produced by these varieties. Some produce more, some produce less. Obviously, commercially, you want more. So let me just start by the top. Most varieties in the gene bank are non-fragrant, and they have almost no aroma. So see, it's less than 0.5, undetectable by the human nose. The BADH2.1 allele, which we've talked about, has an 8 base pair deletion right there in the middle, produces quite a bit of fragrance, and it is found in many different types of rice. Most of the other mutations are found in only one individual that was sequenced, and they are very rare, but each of them knocks out something in the gene and gives some amount of aroma. They come from all over Asia, and the finding really that's important here is that the BADH2.1 allele that has the 8 base pair deletion is the most widespread of any. It was found in almost a third of fragrant rices. So rices from all over the world have the same gene. And so BADH2.1, the one we first diagnosed, is the most widespread, but importantly, many parts of Asia for which we do not have their varieties in the gene bank have fragrant varieties due to independent mutations identified in individual populations of plants in different places across Asia. So there's lots of variation, and there are lots of ways to mutate or knock out a gene. Thailand at the time was hot in pursuit of cloning the gene for fragrance in their most important national rice variety, which is Thai jasmine. And in the nation, they reported, on Wednesday, His Majesty the King applauded Thai scientists and those involved in the patenting of genes that can control the aroma of Thai jasmine rice. His Majesty said the patent would ensure that Thais take pride in eating Thai rice. They won't have to eat rice that has a foreign patent. Many of the scientists involved in that were friends of mine. And I had information that they didn't have because I had looked at the whole spectrum of aromatic varieties across Asia. And in the background, there was another patent pending where the Indian government had brought a case against Rice Tech in the US, which is a US hybrid rice breeding company, for infringement of the name. So as you can see, this is about a mnemonic, the word basmati, and eventually the case was determined in favor of the Indian government, who said, based on the UN's Geographical Indications Act, that the product name basmati incorporates not only the genetics of the variety, but the geographical origin, the place in which it's grown. So much like champagne can only be designated as the wines from the Champagne region in France, basmati can only be used as a term for the rice that is grown in the Kashmir region of India and up into Pakistan, Afghanistan, and over into Iran. So I was sort of embroiled in these controversies of genetics, terroir, as we call it, and national pride. And I had friends that were scientists in India, in Thailand, all over the world. And they would come to me and say, Susan, help us mediate this because you have information on all the rices and we study our national rices. So I set out, it's a group of friends and colleagues, to find out between basmati and Thai jasmine, which we had demonstrated have the same aroma allele, where did it originate? The Thais were on the verge of patenting it, claiming it was of national origin. So there's Indian basmati and there's Thai jasmine. Thai jasmine, Indian basmati is a group 5, as you remember, genetically, it clusters with the group 5, it's a japonica. Thai jasmine is an indica, it's an aromatic rice, it cooks up soft, it's a delicious rice if you haven't tried it. And in order to walk you through this example, believe me, I know many of you will want to close your eyes here, but I just want to say it's pretty cool because this helped settle a dispute. Okay, so what I'm showing you here is there's the bad H2 gene, and this 8 base pair deletion is highlighted by the yellow box with the plus in it, with a minus in it. That's the 8 base pair deletion. So the group of varieties that are japonica, the top row, are japonica and non-fragrant, do not carry the yellow box. The second row is the group 5 basmatis that are fragrant. They carry the yellow box. The third are tropical japonicas. They're fragrant. They carry the yellow box. Then we have the indicas. They're all in red because they differ at every one of these 78 DNA variants, and that's how we know they're the indicas. And they do not carry the yellow box. What we wanted to know was, where does Thai jasmine fit into this? We know it's an indica. And we can see that anything that carries the 8 base pair deletion is fragrant. Indica sequence is clearly different than japonica. The flanking DNA is ultimately what you need to confirm ancestry because mutations don't travel as points. They travel as chunks of DNA with flanking sequence that is carried several other genes at the same time through breeding and reproduction. And here is the Thai jasmine. It's fragrant, it carries the yellow box, and it carries the japonica DNA in the flanking region, but at the far edges, you see it reverts to indica. This is proof that Thai jasmine inherited a chunk of DNA from a japonica ancestor that carried the fragrant allele, and it perfectly matches by these bright green things that it came from a basmati ancestor. So the conclusion based on this DNA evidence was that the fragrance allele and 1.4 megabases of flanking DNA in Thai jasmine was inherited from a basmati ancestor. Now, what does this have to do, and I'm getting to the end, I know, for those who want to protect their national treasure? Well, patent law has been an evolving thing. And as we know it today, patent law allows crop varieties to be protected as inventions because they are unique assemblies of alleles at thousands of genetic loci. Terroir is recognized by the Geographical Indications Act. So you can patent a variety and you can essentially trademark the name and attach it to a geographical origin. But individual naturally occurring genes like the mutation in the BADH2 gene cannot be patented. No naturally occurring genes today can be patented. And it's an important evolution of patent law. All new crop varieties, no matter where they are or how they're developed, derive from those that came before through crossing and reproduction. And the genes are recombined and shared like variations on a theme. And I mention this because I know that many of our speakers in this series often talk about music. And music, like breeding, captures tunes, variants, sounds. The notes are shared, but the conglomerate of the whole is unique. And that's what a variety is. It's a unique recombination of all the genetics, of all the genes and alleles that could possibly be recombined. And we can recognize it as a true invention. So I'll finish up by saying, what's in a grain of rice? Some of you have had a chance to look at those grains coming around. Well, sometimes it's just 2 or 3 layers of a pericarp that gives it, or of a bran color, that gives it a different appearance. And they're highly valued, the red grains, the purple grains, the black grains. And if you cook them as whole grains, they have a very interesting nutty flavor. Sometimes it's the shape. So up here in the right-hand corner, in the US, we have a classification system that recognizes 3 types of rice grown in the US: short grain, medium grain, and long grain. And in fact, they have very different cooking qualities. The short grains cook up sticky. You can eat them with sticks. They're sushi rice. The medium grains are the things that you cook for things like risotto and stews and paellas. And the long, thin grains are the things that cook up as fluffy white rice. So I think there are some fundamental things we all think about when we look at a grain of rice. And I will say that here is along the bottom are many different sizes and shapes, just to give you a clue as to how many there really are. And on the right-hand side here, I show a picture of some hybridization we were doing in my lab, breeding purple pericarp rice with a light hull color. So you can see the pericarp inside the hull, and they're different colors. So my answer to my own question, and you may have different answers, what's in a grain of rice? Well, I see history, I see knowledge, connection to ancestors, livelihood, food, family and community, fertility rituals, unique adaptations to environment, and color, diversity, and highly sophisticated cuisine. With that, I'll end. Thank you very much. Can we take some questions? I know we're over time. Yeah. Oh. I'll hang out in the back. Can we do a couple? Yeah. People can leave if they have to. So is that Peter? It's hard to see. Yeah.
Test, test.
So what's the function of the chemical that gives the aroma? And as the majority of rice, you said, does not have it, what is the advantage of not having it?
It's a question I had wanted to explain, but I didn't have time. In the biochemical pathway, it's an intermediary product. And it's normally metabolized, and it is part of stress resistance pathways. So it does have a function. And when you produce it, bottle it back up and produce it as something, as a volatile that's going to be emitted rather than processed into the forms of stress resistance that the plant needs, the plant is highly stressed. And that stress increases the production of the aroma. So there's a certain interesting feedback loop there. And your question is very well pointed. There are other purposes for that molecule, but when it's valuable, you tend it carefully. And that's how basmati rice is actually grown. Yeah, when they don't have the fragrance, it's because the molecule, excuse me, the molecule is broken down rather than emitted. So it's immediately broken down as part of the biochemical pathway. Ron?
Wonderful, wonderful lecture, Susan. Thank you. My question is just, I ask you this every couple of years. What's going on with the international kerfuffle and controversy over golden rice?
I knew that question would come from you. Many of you heard about golden rice in the mid-1990s. It's a thing. It exists. It is a point of controversy. Nowhere is it produced commercially. It's a GM rice that was used to defend, if you will, improving nutrition, vitamin A particularly, for millions of people who are short on it. But breeding that into every one of the thousands of strains of rice that are consumed by people who need the vitamin A is simply impractical and never took off. So in the Philippines, where they developed it, they tried to subsidize the growing of it. And there are several monasteries in the Philippines that grow it. That's all I have to say. Yes.
The deal with, hold on, what's the deal with, was it arnica? No, not arnica, excuse me. What am I trying to say? That certain rices, brown as opposed to rice, arsenic, excuse me.
Arsenic.
Arsenic,
yes.
that's what
Okay,
I wanted to say.
Good. So arsenic is, of course, naturally found in the soils and in the water of the earth. So very abundant. And in many parts of the world where they've drilled tube wells to access water below the ground, for instance, Bangladesh is the most afflicted by arsenic. The water that they pump up during the dry season, which is coming from the tube wells underneath, is laden with arsenic. And then the rice takes up the arsenic. In the US, the reason the southern US rice growing areas are sometimes labeled as high arsenic is because they once grew cotton on those soils, and arsenic was used to kill the bollworm before BT. Now that we have BT, we don't use any of those kinds of pesticides anymore, but it is kind of important to realize that it can come from, as a pesticide that was applied, it can come from deep within the earth. It has many sources, it's a very abundant. And rice is particularly problematic. It's okay if the rice takes it up into its leaves, but if it puts it into the grain, that's the problem. So again, we did a little bit of work on this. There is a gene that will regulate whether the arsenic is actually just accumulated in the leaves or whether it moves through the transport system and is deposited into the developing embryo. And we have rices that no longer deposited it in the grain. So those are the kinds of, and it's naturally occurring. We have, this is not a GM at all. So there are types of rices that accumulate less. And there are certainly times of the year when it's less problematic. So when there's a lot of rain in Bangladesh during the rainy season, it's not so much of a problem. So all I can say is, yes, it's a problem. And if you're not a rice eater that's eating rice 3 times a day, it's probably not a problem in the US. But for rice eating people, it has become a problem. And you can see the arsenic poisoning on the skin of people throughout the rural areas. And
We have one last question.
So there is rice in it. There is rice in Italy. Where in your classification fits this in? Does it come basically through the Silk Road or on different ways?
So rice was originally introduced to Italy through the Silk Road. When Marco Polo brought rice to Italy, people had no thought about putting it in the ground and planting it. It was this highly prized grain that they liked to eat and look at. And many hundreds of years later, the industry decided to start planting it. And the breeders have then, the Italian breeders have developed rices that are uniquely, I mean, the risotto rice is really not what came from China, but it started with that. And they've interbred with things from all over the world, and they have their own risotto domestic industry now. But the original rice did come from China, and it was Marco Polo. And the risotto is Japonica rice.
Thank you very much. Thank you very much for a great lecture, Susan. And please accept this on behalf of,